Section 8.5 is over translation. The key concept here is that translation converts an mRNA message into a polypeptide or protein. Amino acids are coded by mRNA-based sequences. It's just really the reading of the letters. Translation converts mRNA messages into polypeptides. Remember, that's just a fancy word for protein. And a codon is a sequence of 3 nucleotides that codes for each amino acid. Remember, we talked about methionine. It's the first codon for every polypeptide chain. The genetic code matches each codon to its amino acid or function. There are 3 stop codons. There's only 1 start codon, that methionine. And there are 2 separate ways that we can decode this message. The first is this chart that's provided in your textbook. Some people are going to find this easier to use, and some people are going to find the wheel easier to use. I'm going to show you both of them. The first step is you find the first letter in that 3-letter codon. So let's say it's AUG. Your first letter would be here. So here's my A. The U is in this column, so it's got to be somewhere in this box. And then the G, the third base, is over here. So AUG codes for methionine. Here are the examples side by side. This is what the RNA codon chart looks like, and this is what the codon wheel looks like. The chart, you find the first base, second base, third base, and then wherever that is. The RNA codon wheels, some of you are going to find it's a little bit easier to use. If we were doing CGA, for example, here's my C. So the first letter is in the first circle, the second letter is in the second circle, third letter is in the third circle, and then that gives you what amino acid it codes for. So if we were doing CGA, C, G, and there's my A, we can see that it codes for arginine. Now remember, it's the same information, it's just in 2 different formats. So if we were going to do this and we find our first base, here's my C, and then I find my G in the column, then you're going to find that A. And there it is. Oh, sorry. There's my arginine, so there's the A. See, there's 2 different ways to find the same information. The worksheet that we would normally do in class, but since we can't do it, we're going to do it on here. The wheel at the right shows you how to determine each amino acid goes with which mRNA codon sequence. To decode the codons, start at the middle of the circle and move outwards. So our first example here is AAC. So AAC, and that tells me that's asparagine. UCU codes for, here's my UCU, serine. Now you might notice that that's a big chunk of the wheel, all codes for the same amino acid. That happens in certain places on here. There are different codes for the same amino acid, so different ways to get to the same thing. GAU is aspartic acid, and CCC, proline. Did we get it right? We did. The codon sequences for leucine would be any of the following. So you see there's 4 different ways to get to a leucine, and there's 4 different ways to get to a valine. What are the mRNA's stop codons? Well, you have a UGA, there's a UAA, and a UAG. And then the amino acid sequence, so this is the word that we're forming, this is the sentence. From CGC would form arginine, UAU would form tyrosine, and the AGC would give us our serine. Now, if we were to look at this chart and we were going to do this sequence, the amino acids, it's the same way. GUA, so here's my G's, my U's, luckily they're all valines. My UUU would be a phenylalanine, and the histidine, and then the stop code. Now that's just the beginning, because the next part, some of you are going to really enjoy this. It'll make sense. You're going to take the DNA sequence, and we changed it, so we had a mutation of some sort. The products of transcription and translation would be affected like so. If we had this, and we converted this DNA strand, how do I know it's DNA? Because it has a T in it. Remember, those bases, the only time we have a T is when we're dealing with DNA. It becomes a U when we translate it to RNA. So the mRNA sequence would be a G becomes a C. The next letter is a C, and that becomes a G. The next letter is a T, and that T becomes an A, because remember those base pairs, those rules apply. And then the next letter is an A, and we can't convert that to a T because we're not going from DNA to DNA. We're going from DNA to RNA. So the T becomes a U. And then so forth. We're going to work our way down. The T becomes an A, the C becomes a G, the G becomes a C. Now when you get that translated, you need to go ahead and split up those codons, and we're going to read that amino acid sequence. Okay, so transcription and translation. So that CGA, we're going to go here and find a C, my G's, my A's. And that should give us arginine. It does. And then a UAU, if you look on your codon chart, UAU gives us tyrosine. And then AGC, serine. So that's our amino acid sequence. Now, if we were to come down here and we were going to have that mutation, if you look carefully, you see that the DNA, the only letter that's changed is that T was substituted for a letter G. So when we do the mRNA sequence, it's going to look like that. Now, does it matter in the products? In this case, it doesn't, because arginine is one of those amino acids that has multiple ways to get to it. So that mutation would be essentially irrelevant. It's a silent mutation. It doesn't affect the outcome. So that sequence of amino acids remains intact. There are cases such as sickle cell anemia where a single letter substitution like that is actually going to completely alter the amino acid sequence, and it prevents those people's red blood cells from forming the nice bowl or saucer shape that we like to have, and instead they're like a broken plate. They can't carry oxygen. A change in the order in which codons are read changes the resulting protein. Regardless of the organism, codons code for the same amino acid. This is why those protein shakes that you guys like to take, some of them are plant-based, because a protein is a protein is a protein, just like an amino acid is an amino acid is an amino acid, and DNA is DNA is DNA. It's all the same thing. Amino acids are linked to become a protein. The anticodon is the plug-in end on that tRNA molecule. So the anticodon is another form of RNA that's flipped back. The ribosome consists of two subunits. There's a large subunit and a small subunit. And there are 3 binding sites for that tRNA. So that anticodon is going to fit into here. And then the binding sites, we're going to pull all of those free-floating nucleotides in, and we're going to pull all of those amino acids in. We're going to form this nice chain. And there, clear as mud, right? So translation is going to convert the mRNA transcript into a polypeptide. You can see we come out of the nucleus. There's our mRNA. It's going to find that ribosome. Our tRNA molecule is going to transfer those amino acids in. And that ribosome is just click, 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 making these little chains of polypeptides. And you can see on down here, that one's much further along. Same strand of mRNA. It's carrying the same message. It's like when you guys, one person does the homework assignment and then the rest of you copy it. That's kind of what's going on here. The ribosome helps form a polypeptide bond between the amino acids. The ribosome pulls the mRNA strand the length of one codon. So it kind of chick, 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 chick. This is constantly happening in your body. The now empty tRNA molecule exits the ribosome. The complementary tRNA molecule binds to the next exposed codon. Once the next codon is reached, the ribosome releases the protein and it disassembles. We have another video that I'm going to link. It's one of my favorite. It's also on my website. I'm going to link that video so you guys can see how this is happening. As you're watching the video, you might notice that the little yellow things that are flying in, those are all of the tRNA molecules.